Welcome back, Biology 30 students. This is Mr. MacKay again, going to give you a hopefully brief description of the process of mitosis. I don't want to get too much into it. There's a million videos online you could watch if you don't want to listen to me, which I understand. But here's sort of the big points you need to know. First of all, the stages of the cell cycle, not just the stages of mitosis, the stages of the cell cycle. We start with interphase. All of this going around here is interphase. They say approximately 90% of a cell's life cycle is spent in interphase. An interphase is defined as the time when the cell is doing its normal function. So if it's a blood cell, it's doing its blood function. A muscle cell, a liver cell, whatever, it's doing what it was designed to do. Interphase is broken down into three parts. There's G1, the S phase or synthesis phase, and G2. G1 is a time of growth, as it says, and preparation for DNA synthesis, but again, the cell is doing its normal function. The S phase is DNA replication. The next card I'm going to go into a bit more detail there. And the G2 phase is preparation for mitosis, and again, the cell is doing its normal function 90% of its time. The rest of the cell cycle, or 10% of the cell cycle approximately, is doing mitosis, which most of this video is about. One quick thing, G0, some cells remove themselves from the cell cycle and they stay in the G0 for essentially their whole entire existence. Example here would be nerve cells or cells in the brain that don't really do mitosis, whereas cells like your blood cells and skin cells and stomach lining cells whip through this cell cycle relatively rapidly, within a few months anyway, so we're constantly replacing those cells. We don't replace our brain cells very quickly, which is why things like concussions and stuff can have such long-term effects on an individual, because those cells don't really do mitosis. But let's keep going with the cell cycle. The important part of the interphase is the S phase stage, and that's when the chromosomes duplicate. So right here we have two chromosomes before the S phase. After the S phase, we still have what's considered two chromosomes. So we've got a green, that's one. This is still one chromosome over here, and the purple is the second chromosome. Some important terms, though, you need to know. Once a cell has replicated, once a chromosome has replicated during the S phase, it now has what's called a sister chromatid. These are identical copies of each other. They carry the exact same information. They're basically a photocopy. This green chromosome has photocopied. What we also have, though, are homologous chromosomes. Homologous chromosomes, you get one chromosome from mom and one chromosome from dad. Those chromosomes are very, very similar, but they're not necessarily the exact same. So, for example, maybe your mom gave you a gene to have red hair, and a dad gave you a gene right here to have brown hair. So they both gave you information for hair color, but the information maybe wasn't the same. Or maybe mom gave you information for brown eyes and dad gave you brown eyes, which means it's the same information and it happens to be the exact same trait. So it's very similar, but they're not the exact same for homologous chromosomes. Sister chromatids, again, are identical. And this duplication happens during S phase of interphase. Okay, let's get into mitosis itself. Mitosis has 4 stages. We start with prophase. I should have had that as a heading. Prophase has 5 events that you need to recognize in no particular order. Centrioles form, and spindle fibers form. So centrioles, think of them like the anchor for the spindle fibers, which are technically protein tubules that reach out and are going to grab hold of the chromosomes. Later on in the mitotic stages, we're going to separate those chromosomes that are going to be pulled to the poles. Spindle fibers and centrioles enable that to happen. Some other events during prophase. The nuclear membrane disappears. Nuclear membrane disappears. The nucleolus disappears. And last thing, and this is an important one, chromosomes condense, which means they go from being long and thin to being very, very short and fat. And I always use the example in my classes. Think of them before prophase as being like long noodles of spaghetti, and after they condense are now very, very short noodles like macaroni-sized noodles. That helps them separate much easier without splitting apart. That's prophase. Metaphase, much, much easier. There's only one thing you need to know for metaphase. And in metaphase, all the chromosomes line up at the equator. They line up at the equator. Hopefully you're taking notes, or at least going through your note packages if we're going through these. These are the high notes, the things you have to know. So metaphase, straightforward. Next we have anaphase. And anaphase, there's only one point you need to know. Here the chromosomes separate. And to be even more specific, it's the sister chromatids are separating. The sister chromatids separate. One goes to one end of the cell, one goes to the other end of the cell, and you can see that it's happening in all of those sister chromatids that were formed earlier. Lastly, we have telophase. And in telophase, there's again 5 events you need to know, but they're basically the opposite events of what happened in prophase. So in prophase, the centrioles formed. Here, the centrioles disappear. The spindle fibers disappear. The nucleolus reappears or forms. The nuclear membrane reforms again. And the chromosomes uncondense. Chromosomes long and thin again. Long and thin. And then we have one last important thing that happens during telophase called cytokinesis. And cytokinesis is what you see happening right here, where the cell's pinching off and dividing and separating all the cytoplasm, all the stuff that's in this area, all the different cell parts, the ribosomes and the endoplasmic reticulums and all the different parts of the cell that we learned about in like grade 7. Those things separate so that at the end of telophase, this completely breaks apart and we have 2 brand new cells. And the key thing with mitosis is you get exactly what you started with. So I'm going to back up on my cards here for a second and look at our chromosome number. In this cell, we have a chromosome number of 4. 1, 2, 3, 4. So it's said that it's diploid, or 2n number equals 4. At the very, very end of mitosis, we're going to have 2 cells that also each have a diploid chromosome number of 4. That's the key with mitosis. You get exactly what you started with. And that's super important as opposed to meiosis, which we'll learn about later, where the number gets cut in half. Mitosis, the number stays the same. Hopefully that was short enough that you paid attention to the whole thing and took some notes down. Watch it again or pause it wherever you need to. And certainly feel free to email me any questions or things that aren't clear. Talk to you later. Bye.